Hey, yeah, good afternoon, everyone. Thanks, Ron, for the great introduction. So in the next 20 minutes or so, I will take you through the journey of the CliniMACS platform, which started its groundbreaking start 25 years ago. And over the years, I will highlight the continuous innovation that continue to push boundaries for the therapeutic cell manufacturing. Okay, since this is the anniversary event, it's only fitting to start with a little bit of history. So it started 25 years ago with the introduction of the CliniMACS Plus instrument, which really changed or became the standard for the clinical cell separation. So and it became a standard because it offered functionally closed and automated way of separating the cells. So it received the CE mark as a medical device for the first application for isolation of hematopoietic stem cells back in 1997. But since then, the number of applications grew, and many others, it extended far beyond the hematopoietic stem cells with CAR T cells, monocytes, B cells, NK cells, and also Tregs. Yes, but also this device is now well accepted in the field. There are 50 countries where it's covered from the regulatory standpoint. There are all the devices deployed globally performing tens of thousands clinical cell separations as we speak. And there are 7 commercial stage cell therapies where this device, CliniMACS Plus, is an integral, critical part of the manufacturing workflow. But of course, we didn't stop there. So 10 years ago, we introduced the next step in the evolution of the CliniMACS platform, CliniMACS Prodigy, the device that, besides the advanced cell selection capabilities, it also has the centrical unit, which allows for the cell washing or transduction and cultivation inside the same platform. So it really and besides that, it also has the gas mixing unit, which allows the users to define the parameters, to set up the parameters for the cell culture they are running. So this device redefined therapeutic cell manufacturing because it offered fully automated and closed system, at the same time offering the flexibility, allowing it to be adapted to very specific use cases, very specific applications. And this device, CliniMACS Prodigy, it's also now well accepted in the field, widespread. It's used in close to 200 clinical trials, and it's also an integral part of cell therapy manufacturing, so it's part of the 6 commercial stage cell therapy products. But of course, we are not only the instruments company, we are the whole workflow company. So it's our philosophy to offer our users complete workflow, instruments, consumables, reagents, starting with buffers that are used to dilute the starting material, that are used throughout the cell separation, and the reagents for activation, for transduction, for expansion, and also for the final formulation. So this kind of holistic approach ensures that our users, our customers, get seamless and efficient cell manufacturing process. One of the recent innovations is that we worked on was to increase the culture volume. This is to target the applications, clinical applications, which will benefit from the larger cell numbers. This is, for example, the TCR-T applications or applications of therapeutic T cells targeting solid cancers. But at the same time, the larger culture volume and the larger cell numbers allows for certain efficiency and flexibility that can also be used to shorten the manufacturing process. I will talk about it a little bit later. So 2 years ago, in 2022, we've launched the new application, T-cell transduction large scale, or TCT-LS as we call it, which uses the larger cultivation chamber, and that allowed it to bring the final cell numbers, to increase it almost threefold and to bring it to anywhere from 14 to 21 billion cells at the 12 days of manufacturing. And this, for the majority of clinical applications, is the therapeutic dose that will be enough. So this is the, to showcase, this is the case study published by Miltenyi together with GSK. This is the study about developing the manufacturing process for the TCR-T asset from GSK on the Miltenyi manufacturing platform. So as you can see, maybe you can see, I hope you can see here in the graphs, that this process was consistently able to manufacture 1.5 times 10 to the 10 total cell numbers in the end. This is much higher than our regular process with which we are comparing on all these graphs. But at the same time, this increase of therapeutic dose, increase of the final cell dose, that didn't disrupt any process. So we achieved the cells exactly in the same quality as we used to in our regular TCT process. So the, for example, CD4-CD8 ratio was completely comparable to our regular size process. It was the same, cells were of the same, almost 100% viability at the end of the culture, and also the purity and yield was similarly high to the standard TCT process. And maybe most importantly, the cells still preserved the TSCM or memory stem cell phenotype, which is the most predominant population at the end of the culture, similar to our standard TCT application. That ensures that the cells will be still able to proliferate and to engraft when they are injected in the patient. So the next few slides will guide you through the NPM1 story. So NPM1 or nucleophasmin-1 it's a gene which harbors recurrent mutation in AML, acute myeloid leukemia. And this mutation is it's a frameshift mutation. So as a result, the new peptide is being translated, which is also presented by MHC class 1 molecules. So this makes it a strong target for TCR-T therapy. And that's exactly what we've been working on. So this is our manufacturing process for the NPM1 specific TCR-T CD8 positive T cells. So it's an 8-day process. It starts with frozen leukapheresis, which is collected about 15 days before to allow for patient conditioning. And then we select CD8 only cells in this case, and we seed 2 times 10 to the 8 cells in the chamber, activate them. On the next day, they are transduced with lentiviral vector encoding the TCR specific for delta NPM1, for NPM1 mutation, and then the cells are being cultured in the large chamber in the TCT-LS large-scale application. So we're doing multiple. Oh, you can see my mouse. So we're doing multiple QC at different days, but the last point is day 7 when we assess if we have reached the target dose, which is 1.2 10 to the 9 TCR positive T cells. If we haven't, this is a flexible process. We have the possibility to extend the manufacturing until day 10, but in most cases we have more than enough cells, so the product is formulated at day 8 as a fresh product ready for injection. And you can see that viability of the cells, it's very high, almost 100% at day 8. And in the end of the process, typically we get around 8 billion total cells with a transduction efficiency of around 50%. That gives us 4 billion TCR positive cells, our effector cells. This is far exceeding what we need, 1.2 billion. And you can also see on the right graph that the resulting cell product is extremely pure. So this is pure CD8 T cells, which ensures that they will be therapeutically efficient when they will be put back into the patient. So and the cells are active in both in vitro and in vivo studies that we've done with them. So you see that in vitro, they respond to the stimulation with the target cell line. They respond by secreting cytokines. They respond by killing the target cell line. In vivo, they are able to to control the tumor, which shows that they are fit for purpose and they indeed have the therapeutic potential we were looking for. As we speak right now, the clinical trial is being started. I think first patient is about to be injected, if not already. And also we, over the last few years, we've introduced a number of accessories to the CliniMACS Prodigy platform. This is the workbench, the electroporator, and the CliniMACS formulation unit. I want to dive deeper now to the CliniMACS formulation unit, which allows for the automated aseptic, gives us an automated aseptic process for formulation of the drug product in combination with CliniMACS Prodigy, of course. So you can see this formulation unit at the top as this green bar, which is connected to the Prodigy. It is connected to the Prodigy and it is controlled by the Prodigy. It enhances the Prodigy by giving it additional 13 pinch valves. In combination with the formulation unit, we have also developed and released a tubing set with the number of bags in different sizes for QC and for product formulation, either fresh or frozen. And also very recently we've introduced another formulation set, which is called a flexible formulation set. And this has 10 lines which users can use whatever they like for. They can attach themselves the bags of their choosing there. So together with the tubing set and the formulation unit, it transformed the Prodigy platform into the comprehensive end-to-end solution, starting with patient starting material and ending up with a drug product ready for injection. And this brings us to more or less today. So this second part of my talk may be more interesting to you. This will be focused on the upcoming improvements and future applications which are still in the development pipeline. So what I shown previously is either published or released. What I will show next is what is in our pipeline. So that's what we are working on right now. So the future improvements that I will show today, they're focused mostly in the three directions. So the continuation to improve our end-to-end process, the shortening the manufacturing process, and last but not least, by developing new applications for the new cell types, which can broaden the scope of the clinical applications. So first is our efforts to continue to develop end-to-end process. So we have developed two new tubing sets which bring together the core part of the tubing set and the formulation part of the tubing set, which I showed you a couple of slides before. So this also it is reflected on the side of the process, the application. So the workflow also is now one workflow from start to finish. So those two tubing sets, they're based around our regular size and our large size chamber. And the large size chamber is able to give you the target cell dose of about 20 billion cells. In addition to that, the tubing set with the large size chamber, it also has a larger column, which allows for a single stage separation of up to 4 × 10⁹ labeled cells. And so together this really even further pushes the boundaries and creates the complete end-to-end solution that will serve the needs of the customers who want to stay on a single platform. Okay, to continue, I will shift to the short manufacturing. So short manufacturing has several advantages. On one side, there is a possibility that the cells which have been in culture for a shorter time, they are less differentiated and they will have better expansion, better persistence once they are injected into the patient as a drug. On the same time, definitely the shortening the process reduces the vein-to-vein time and it gives the patients so much needed therapy sooner. Additionally, it allows for the more efficient use of the resources at the manufacturing side and thus lowers the cost of manufacturing. And even if you have a flexible process, as I've showed you with the NPM1, there is also a possibility to do better planning, for example, around the holidays. And altogether, those improvements, they really make use of more of the same manufacturing facility with exactly the same infrastructure. They make better use of it, and so the facility can manufacture more products in the same amount of time. So the software which operates the application, it's our software by default is doing the 12-day process, but in reality it's flexible enough. So users can themselves modify the activities in the activity matrix. They can change at what time the activities are being performed, and they can also shorten the expansion phase to fit for their applications. So the data that I will show you next was done on our off-the-shelf standard TCT application, in which we modified the activity matrix. So we used the application to start with the fresh starting material, and we manufactured the fresh drug product. We used two independent CAR constructs, and we modified the activity matrix so that the whole process will take 7, 8, 10, or 12 days. So as you can see here, so the two constructs are left and right, CAR1 and CAR2, and you can see that the expansion curves for the processes with a different duration, they really superimposed. So by day 7, we are reaching approximately 2.5 times 10 to the 9 total cells. By day 8, it's increased to 3.7 billion cells, and at day finally at day 10 or 12, we're reaching the maximal cell numbers for this chamber. It's 5 billion total cells. And you can also see that the viability starting from day 5, it's almost 100%, and it consistently stays at 100% level, or close to 100% level. You can also see that the cell composition that at day 7, at day 8, day 10, and day 12, we have almost only T cells in the drug product, so it's 96-98% T cells, and that the CD4-CD8 ratio, it's relatively stable over these time points that we interrogated. And with the transduction efficiency also is stable after day 5. So for CAR1, we are having approximately 50% transduction efficiency. For CAR number 2 on the right, it's around 30% transduction efficiency, which gives us, for both constructs, at day 7, around 1 billion target cells, which is more than enough for most application for CAR T cells. And those cells, they are active, they are in vitro, they recognize the target cell lines, they are able to secrete cytokines, they are able to perform lysis of the target cell lines. And interestingly enough, the cells that were cultured for a short period of time, 7 days, they secrete more cytokines, in particular IL-2 and interferon gamma, compared to the cells that were manufactured for longer times. And also, finally, we have had to confirm that the cells are active also in vivo, and we saw that indeed they are both cells manufactured at 7 days and also our conventional process 12 days. They are both capable of eliminating the tumor from the model, mouse model, and this confirms that the cells manufactured at 1 week period, they are completely fit for purpose. They can be used in the clinical setting. In parallel to us working on making the process shorter and to show that the cells are still capable of performing their clinical tasks, our customers have been using the flexibility of our platform to do the same, to explore if cells can be manufactured in a shorter period of time. And yes, they found the same. So cells can be manufactured it was featured in several publications that cells on the Prodigy can be manufactured from 7 to 9 days. It was described in both preclinical works describing the process development, but also in publications featuring those cells being used in the clinical trial. Okay, and finally it brings me to the last part of my talk. I would like to talk about TILs, tumor-infiltrating lymphocytes, and the process that we've developed on the CliniMACS Prodigy to manufacture these cells. So TILs, of course, they have they're really well suited to tackle the solid tumors. They have, by nature, they are polyclonal. They have polyclonal TCR repertoire, so there's the risk of tumor evading is lower because they attack multiple antigens at the same time, and also they are autologous, and they have natural T-cell receptors, so the risk of adverse side effects is minimal with them. But at the same time, the manufacturing process has lots of challenges. On one hand, the cells, they come from the immunosuppressive tumor environment, so they are exhausted already from the start. On the second hand, the conventional manufacturing process for TILs, it's really complicated, it's long, it takes several weeks, and also it involves a lot of manual steps and a lot of open handling steps. So in order to streamline this process and to bring the benefits of automation to that, we've developed what we call a tumor-reactive T-cell or TRT process for the CliniMACS Prodigy. This is a really flexible process which reflects that the TIL companies and TIL academic groups working on TILs, they're really doing it in multiple different ways. So we wanted to accommodate them with our process. So this process can start with different starting material. It can start with tumor digest. It can start with outgrown TILs. It can even start with peripheral blood. Then there is an optional antigen stimulation step on directly on the Prodigy within the same process, which is followed by CD137 enrichment step. Then the cells are activated by adding feeder cells and the agonistic CD3 antibody. They are polyclonally stimulated. And then after that, there is another optional step of the transduction. So if the customers want to put certain genes in their TILs, that's also possible within the same process. And finally, the cells are expanded on the Prodigy in the rapid expansion protocol, which were transferred to the closed and automated system. So the process is really flexible, that it can be tailored to meet specific needs of specific customers, and it offers various entry points that can be adjusted to different requirements. You can see here that this is the kind of validation of the automated rapid expansion protocol on the Prodigy. In the automated setting, we've used the outgrown TILs from different tumors, from melanoma, from pancreatic cancer, from renal cell carcinoma, colorectal carcinoma, and also breast cancer. And you can see that even when we started with not very high number of cells, below 1 million cells, we got to after 15 days of the cultivation, we got the cell dose of approximately 4 billion TILs at the end. But of course, we understand that certain TIL applications would require even higher numbers. So we've adapted the large chamber, of which I've spoken a few slides ago. We've adapted it also to the TIL process. So this is still in the making. This is still in the pipeline, but we are working on it right now. So here we can see that with the large chamber, we can increase the dose. At the end of the 15 days, we can get to as much as 20 billion cells in the Prodigy. And also, despite this increase of the cell numbers, the viability stays as good as it is on a smaller process, so it's always higher or equal to 95%. At the same time, as I mentioned, the Prodigy with the magnetic column allows for to really enrich for tumor-specific cells. So it was reported in multiple studies that the TILs that are expanded in the conventional process, the majority of them are not really tumor reactive, so they are just bystander cells which are being expanded in the process. At the same time, we can use the fact that when seeing their antigen, when being stimulated through the TCR, T cells express on the surface activation markers such as CD137. So in this case, we can stimulate the cells with the tumor antigens and select downstream select only the specific cells and then expand only the specific cells, increasing the potency of the therapeutic product. So this is to demonstrate you how it works. So the cells are incubated in the Prodigy for 24 to 48 hours with the antigen. It can be tumor digest. It can be even the synthetic peptides covering tumor new antigens from the tumor, and we are getting the out of this process. Afterwards, downstream, we separate the cells. We select the CD137 positive cells with the GMP grade reagent, and we can see that we're getting the from 80 to 99% purity of the CD137 positive cells. And as a proof of principle, we expanded cells from 6 different donors, which was selected for CD137 positive cells. So they do expand in the Prodigy. If we start with the recommended cell numbers of 1 million, we get to the 4 billion cell as usually. This is, to remind you, this is a regular size chamber. If we start lower, sometimes we also can reach the target cell dose of 4 billion cells. And regardless of the starting cell numbers, we can see that the viability of the cells are close to 100% at the end of the process. So this is the end of my talk. I just wanted to emphasize here that the progress of the CliniMACS platform it's extended far beyond our internal efforts. So in parallel to our work, our customers in the field, they're using the flexibility of the process of our platform to explore, to advance their therapeutic applications. They're developing cells to treat solid tumors, to treat autoimmune disease. That they're developing the manufacturing processes in the point-of-care setting. And as I said, they are working to, as we as well, where they're working to independently to shorten their manufacturing process. They are also exploring the boundaries. They're moving outside of pure autologous space to allogeneic space, and they are also using donor-derived cells in the context of allogeneic bone marrow transplantation. Though this altogether highlights that this widespread innovation by our customers highlights that the platform is versatile and has great potential. So with that, thank you for your attention, and will be happy to take some questions now.